Alrighty, so let's go ahead and get started with the lecture on microscopic organisms. I'm just making sure I'm recording. And here it is. Okay, so this is chapter 8 again. The first topic on biodiversity, and again, specifically on microorganisms. Right, so these are the topics that I'm going to be covering, starting off with the origin of life, an overview of prokaryotes, archaea, bacteria, differentiate between the two, DNA transfer and genetic variation in bacteria. A little bit about eukaryotic origins, so the how eukaryotes originated. Protists, the first signs of multicellular life, viruses, HIV, and then ending off with prions. All right, so let's go ahead and get started with the beginnings of Earth, right? So early Earth was an inhospitable place, right? So it was very hot and it was full of gases, okay? And we're saying about 4 billion years ago. All right, so Earth is about 4.7-ish billion years old. And in the beginning, it was evidently really, really hot and an inhospitable place. So nothing could live, nothing could develop, no life could develop on Earth at those times. Okay, but then life actually could develop as soon as Earth started to not only cool down, but also as soon as it started to generate the biomolecules necessary for life to take place. Okay, so as soon as those molecules were generated naturally, evidently on Earth, then life could evolve from that. Life could be formed. So that process is called biogenesis. Okay, so bio, life, genesis, making, starting, and creating, and so on. Okay. Now there's been a lot of experiments on this. Okay, so starting off in the 1930s, and then in the 1950s with this very famous experiment by Urey and Miller that essentially demonstrated that organic molecules can form from simpler molecules such as ammonium, right? Methane, hydrogen, water vapor, heat, electricity, right? So these were the early ingredients, if you will, that their combination was able to form these biomolecules, right, that had never existed before, right? So it's thought that about 3.6 to 3.9 billion years ago, the first signs of life originated. Okay, and that was because of the fact that these biomolecules were being formed. So these two scientists, Urey and Miller, demonstrated that these biomolecules, these larger polymers, could form from smaller molecules. Okay, so these are the smaller molecules. These are the molecules that were thought to have existed before life, or actually 4 billion years ago on Earth. There was water, evidently, that was brought in by asteroids, and these were also brought in by asteroids. Okay, so now as soon as these biomolecules formed, the first signs of life shortly after that, and by shortly I mean millions of years, but keep in mind that in relation to the age of the Earth, which in relation to the age of the universe is nothing, right? The universe is about 13 billion years old, and Earth is about 4.6, 4.7 billion years old. So life, right, is, or actually millions of years, is nothing when compared to the to billions. So keep in mind, 100 million is actually a billion. I mean, 1,000. I'm sorry, 1,000 million is actually a billion, right? So so it, those numbers are relative, obviously, but millions in relation to billions is evidently almost nothing, right? So unless you have enough. So again, biogenesis, right, allowed for first the formation of these molecules and the formation of what we know of nowadays as self-replicating molecules like RNA and DNA that started to be made in self-contained protocells. Okay, because the first signs of life are nowadays known as protocells. So they were these vesicle-like structures which were bound by a membrane, right? So you can see something that looks like cells in here, right? So we refer to these as protocells. I highly suggest you guys look that up. Harvard University carries out a lot of experimentation on protocells, and they've managed to prove that these were in existence way back when, right? So 3.6 billion years ago. And again, we can form these in the lab, right? So we can, by using very simple substances and blending them together and using heat in a pan, if you will, you can form these, right? Again, it's pretty simple to do, but the thing is, you won't be able to see them unless you have a microscope, right? So you'll need a microscope to do that. So in a BSL-1 lab, such as the one that we have at the college, we can actually observe these protocells there. So again, these were the first signs of life, after which RNA, self-replicating molecules like RNA, was actually given rise to, and their interaction made, right, the first prokaryotic cell. All right, so as you know, prokaryotes are very simple and very small cells, which are mostly unicellular. Okay, so all prokaryotes are unicellular organisms. So for the first billion years, right, after life actually came about, the first, again, type of life was prokaryotes, right? So for a billion years, prokaryotic life actually dominated Earth, right? So again, we've gone through this description before, or we've already grouped prokaryotic life before into two domains. One is the bacteria, the other one is the archaea, right? So the domain bacteria is actually found everywhere. So these are bacteria that live in you, on you, and around you, right? And you interact with these bacteria on an everyday basis. Okay. So again, the domain bacteria. So under this domain, we have two kingdoms. We have eubacteria and archaea bacteria, right? So archaea is essentially the same in terms of how many cells they have, because they have one cell, so they're unicellular. Then the thing is that these bacteria tend to be extreme. What do I mean by this? Well, what I mean is that they live in extreme environmental conditions, like highly acidic environments, very salty environments, right? Very hot, very cold environments. That's basically what I mean by extreme environment. So if you were to place one of these everyday bacteria in a salty environment, right, would they survive? Well, not necessarily, right? Again, they have different adaptations that allow them to survive in different environments. So we've seen prokaryotic structures before, so I'm only going to touch on this real quick. Keep in mind that flagella cell wall capsules, these are specific to prokaryotic cells. Flagella are for motility, cell walls for protection, capsules for protection as well, but also for attachment to cells and to surfaces, right? And that, keep in mind, that allows them to form biofilms, right? So prokaryotes do have endospores, as was already mentioned. Endospores in previously. So endospores are thick-shelled protective containers, if you will, that have or structures rather, that have DNA and RNA inside of them. So in case the bacteria face harsh conditions where they cannot thrive, so they're going to go ahead and produce endospores, release them into the environment. And this endospore will ensure that the bacteria survives once environmental conditions are okay for the bacteria to thrive again. Okay, and then they can exist for thousands of years, right? Just waiting, right, for that environment to become better again. So keep in mind that prokaryotes reproduce by binary fission, which is a very simple and efficient way for cell division. Okay. Bacteria come in 3 different shapes, as was mentioned previously. There's cocci, or spherical bacteria. These spherical bacteria can form chains, right? There's also bacilli, which are rod-shaped bacteria. And then there's spiral, or curved, if you will, bacteria. Some bacteria can actually carry on with photosynthesis, and others can actually use inorganic molecules like sulfur, right, or atoms rather, like sulfur, to be able to make their own food, right? So there's these photosynthetic bacteria called cyanobacteria, which are thought to have contributed to the tenfold increase in oxygen, right, at some point during the geological record. Okay, so that oxygen, quote unquote, oxygen revolution, I suggest you guys look it up. It actually contributed to the development of eukaryotic multicellular organisms. Okay, so again, as mentioned, prokaryotes can form biofilms by the use of their capsules, right, that allow them to stick together. So biofilms are very thin layers of bacteria that form on tissues, on objects, and on surfaces, and so on. All right, so now archaea bacteria come in 3 forms. There's methanogens, halophiles, and thermophiles. All right, so methanogens emit methane gases. Okay, so they're gonna go ahead and be smelly, if you will, right? So you're gonna find them in places where there's a lot of trash, for example, but also under thick layers of mud, right? Or swamps underneath. So again, they actually emit methane gas from their metabolism, right? As they ingest and carry on with their metabolic physiological functions, right? They go ahead and release a methane waste product. Okay, so those are methanogens right there. Halophiles love salty environments. They cannot survive anywhere else, right? So I'm showing the Dead Sea right here. It's not a sea, it's a lake, right? But it's highly salty, if you will. There's a lot of salt in this lake. Okay, so we have found bacteria, right, living in the Dead Sea and thriving in the Dead Sea. Again, you put that bacteria anywhere else, it'll die right away. Okay, so it needs that. And that talks to you about their physiologies, right? The ways in which their physiologies work allow them to deal with highly salty environments. Okay, so now thermophiles are another type of extreme bacteria, or archaea. Thermophiles thrive in heat environments, so very hot environments, areas with really high temperatures where water is naturally boiling, right? Such as in springs. Also under the ocean, in volcanic vents under the ocean, right? Very hot environments. You can also see those types of bacteria. Now, the physiological characteristic that allows bacteria, these thermophiles, to thrive in hot environments is the fact that they contain heat shock proteins, right? So they can actually be exposed to eye heat, and those heat shock proteins, again, are adapted for them to be able to survive heat stress, right? Now, heat shock proteins are highly conservative. We have heat shock proteins in our bodies, in our cells. Okay, so heat shock proteins allow us to also resist high levels of high temperatures, if you will, right? Up to a certain extent, obviously, right? Our natural temperature is about 98.6 degrees Fahrenheit, but we can go up to 106, 107 and still survive, right? Now, don't get me wrong, right? Anything above 99, anything above 100 is considered fever, right? So if you do have a fever, then evidently there's an infection going on, right? What I'm saying is, people don't die because of fevers necessarily, although sometimes some people do, right? But again, the fact that we can actually go up into the low hundreds in terms of temperature, body temperature, is in part due to the fact that we have heat shock proteins, okay, which allow our cells to withstand those high temperatures. Now, not to be confused with the reasons why we go into fever, right? We go into fever because there's an infection. There might be a bacterial or viral infection that managed to penetrate our body. But then, even though that's a causative agent, the actual physiological process that causes fever it actually starts in the hypothalamus, right? The hypothalamus is a brain region, which is also known as the body's thermostat, if you will, right? So there's a nuclei inside of a hypothalamus that allow us to regulate body temperature. Okay, so there it is. And again, heat shock proteins are physiological proteins that allow us to be adapted, if you will, to high temperature environments. Actually, heat shock proteins, or HSPs, are involved in exercise physiology, exercise adaptations, and so on. They have a wide range of physiological benefits to the body. Okay, I highly suggest you guys research HSPs. I follow this physiologist, right? Her name is Dr. Rhonda Patrick. I highly suggest you guys look into her work. Look her up online. There's a lot of videos on YouTube. She talks a lot about physiology, nutrition, exercise, but again, in terms of physiologies, right? So the ways in which we can achieve maximal performance, if you will, but through a physiological aspect. Okay, so again, bacteria thrive in most Earth's habitats. They live in you, on you, right? In our gut, right? They live on our skin. Now, they're limited. Bacteria are limited inside of our body, right? They're only allowed to be in our gut and also in our mouth, if you will. So areas, regions within the body, cavities that are exposed to the environment might have some bacteria on them, right? So our intestines have a lot of bacteria, okay, and they help us in producing vitamins, right? They help us in digesting and creating waste products like feces, for example. They have a lot of benefits to us, but they're, again, limited to areas that are exposed to the environment, right? Like the digestive tract, starting in the anus, rectum, large intestine, not the small intestine, because the small intestine is meant for other things like nutrient absorption, right? Not in the stomach, because the stomach is very acidic, right? But if you look in the mouth, right? The mouth, if you analyze bacteria in the mouth, then you're going to find many different species of bacteria. On our skin, we have a lot of bacteria as well, and they're beneficial for the most part, but sometimes they can become infectious and they can cause infections. And disease, if you will, right? So those bacteria that cause disease are known as pathogens. So environmentally, bacteria are important because they're nitrogen fixers, meaning that they help plants absorb nitrogen by fixing nitrogen in the soil. Okay. So sometimes plants are not able to absorb nitrogen naturally, so therefore they need the waste products of these nitrogen fixing bacteria so that they can actually absorb the nitrogen that they need from the soil through the roots. Bacteria are decomposers, as you know, right? So they decompose dead organisms. And that's important because dead organisms, as they break down, they actually become nutrient, right? They become nutrient to plants, right? So when we die, right, there's this philosophical idea, if you will, that when we die, we become, something else, if you will. Well, you can, if you delve right into this idea right here, into this theory, then you can say, if you will, that we do become, but something else, right? But not necessarily spiritually. We probably become something else because we are decomposed by bacteria, and plants actually utilize us as their nutrients, right? And I'm saying us, but I'm talking about animals, right? All animals, and also plants, right? So hopefully that makes a little bit of sense. Sorry about my kids yelling in the background. But yeah. So bacteria, again, we have many uses for bacteria. Many different uses, including in sewage treatment, right? So bacteria, microbes, can decompose sludge and help us to recycle nutrients. We can use bacteria in bioremediation, right, to remove pollutants from the environment. For example, when the BP spill occurred in the Gulf, right, and this occurs all the time, okay, but the BP spill was a major, right, a major issue for the Gulf of Mexico, right? So one of the biggest ones that has occurred in modern history, if you will. They use bacteria. They released genetically modified bacteria, so that those bacteria could, remove a lot of that oil product that was spilled by British petroleum. Right. So what happened was that one of the major pipes, which carried oil under the sea, it actually, I'm not sure if it was opened on accident or it probably burst. So it burst and it was opened, right, and it released a lot of oil into the Gulf. Hopefully you heard about it. You know a little bit about it. If not, then feel free to do some research on that. Again, one of the major environmental disasters in modern history. Again, pathogens are bacteria that cause a disease and serious illness and can cause death in animals. Some examples are Staph aureus with skin infections, right, like scalded syndrome, Bacillus anthracis with anthrax, and Salmonella with food poisoning, right? So this causes mostly digestive infections, right? Lyme disease and Borrelia and so on, right? Just kind of repeating, bacteria reproduce by binary fission, and they manage to achieve genetic variation not through sexual reproduction, because binary fission is actually asexual reproduction, but they still need to be genetically variable, right, in order to ensure survival on Earth, right? And this is why they've been so successful, and they've managed to make it through thick and thin, if you will, right, throughout Earth's history. So transformation, transduction, conjugation, okay, and also plasmid transfer, all of which I've already touched on before, but let me get into a little bit more detail on each. First and foremost, transformation allows living bacteria to absorb DNA content of bacteria that died, right, previously. So as bacteria die in their environment, they release DNA, which is taken up by other bacteria, therefore they become transformed. Why do they become transformed? Well, because the newly incorporated DNA, which came from other bacteria, is actually incorporated and integrated into the genome of the living bacteria, right? So that's a form of naturally genetically modifying yourself if you were a bacteria, right? So now we criticize genetically modified organisms. In reality, it happens naturally, and you're looking at it here, right? So it doesn't happen naturally in humans. It doesn't happen naturally in larger animals, but in nature, you see genetically modified organisms, which is very helpful for the survival, right, of the species. Now we can get into a lot of issues here, but this lecture is evidently not the place for that, right? So another form of DNA transfer that achieves genetic variation in bacteria is transduction. All right, so in transduction, bacteria are actually infected by viruses. And these viruses are called bacteriophages, right? Bacteriophage is a virus. Collectively, these viruses infect bacteria only, right? So as they infect the bacteria, they dock on the surface of the membrane of the bacteria. They inject their DNA, and guess what? The viral DNA becomes embedded onto the DNA of the bacteria. And then eventually, right, that achieves naturally a genetically modified organism, which is genetically variable, okay? Last but not least is conjugation. Conjugation refers to a type of genetic variation process that occurs in bacteria through a sex pilus. Okay, so a sex pilus, we're looking at it right there. It's kind of like a bridge that is formed from one bacteria to the other, right? Through that bridge, they manage to exchange genetic information. So genes are exchanged like that. All right, again, not only does that provide them with genetic variation, but also with genes that allow them to achieve protection against invading microorganisms and so on. The other one is plasmid, right? A plasmid transfer. Keep in mind that plasmids are smaller pieces of circular DNA inside of bacterial cells. All right, so they can be transferred from one bacterium to the other. And once there, they can be duplicated as well, right? So once they're duplicated, then the duplicate is transferred to other bacteria and so on. So these plasmids, keep in mind that they're DNA, right? They contain genes. And for the most part, these genes provide protection to the bacteria, okay? Like, for example, resistance to antibiotics, right? So most bacteria that are resistant to antibiotics achieve that through the use of plasmid transfer. Excuse me. Now, I'm not going to spend a lot of time on eukaryotic membranes because I've already talked about eukaryotic membranes. Let me just mention a few things here. I mean, eukaryotic cells, sorry. So something that's very important for me to mention, right, is the fact that eukaryotes originated because of something called endosymbiosis. So this was an event that happened billions of years ago, right? And what this allowed life to do is endosymbiosis allowed life to take a sharp turn in terms of evolution, right? Why would that be? Well, because of the fact that endosymbiosis led to the generation of eukaryotic cells. Okay, so the way in which it happened, it was first by essentially an infolding of the membrane of these protocells, of these early cells. Okay, so this is an ancestral early prokaryote. There were infoldings, right, in their membranes, which allowed the formation of the endomembrane system, which we've already talked about. It refers to the nuclear membrane and the endoplasmic reticulum, which essentially makes a eukaryotic cell, right? Now, after that, there were events that allowed for the ingesting, right, of these oxygen metabolizing bacteria. Again, they were ingested, if you will, in these processes that took millions of years. And then ultimately, chloroplasts were also ingested by other populations of these newly formed cells. And guess what? Their interaction was positive, right? So positive that the newly oxygen metabolizing bacteria and its ability to produce ATP, guess what it became? It became mitochondria and it became a chloroplast, right? So that essentially created that diverged essentially into what we know of nowadays as a eukaryotic cell, right? And being able to make a lot of ATP with these two organelles, then that allows you to become a large organism, right? So multicellular organism. So am I saying that the mitochondria and the chloroplast were once bacteria in and of themselves? Yep, that's correct. So mitochondria were bacteria billions of years ago, right? And they were, again, oxygen metabolizing bacteria that were ingested, right, engulfed, if you will, by early prokaryotes in order to form these symbiotic mutualistic relationships where they both helped each other, right? The prokaryotic ancestor here provided shelter to this oxygen metabolizing bacteria. And then the mitochondria provided ATP, right? So wasn't that a mutualistic relationship right there, right? Same thing with the chloroplast, right? Now, what are the implications of this? Well, that means that mitochondria and chloroplast, they contain their own DNA. They contain their own genes. They make their own proteins, right? They can divide on by themselves, right? Aside from the fact that they can produce large amounts of ATP, which is energy. Okay. So again, I'm not going into unfortunately a lot more detail on that. I wish we had time, right? But again, feel free to do your research on this and delve as much as you can. Again, very important events that led to the formation and evolution, right, of eukaryotic organisms. Now, protists are other single-celled organisms, although they can be, I mean, they are eukaryotic. Right? That's the difference between protists and bacteria. Right? So these are eukaryotic cells, and these evolved 2 billion years ago, right? So what I wanted to say is they can be multicellular, but mostly they're found as unicellular beings. All right. So basically, anything that doesn't fit anywhere else, if it's not an animal, if it's not a plant or fungi, and still a eukaryote, it's considered a protozoan, a protist. Okay. These are some examples. Paramecium, for example, with their cilia, Giardia, which is an intestinal parasite, right? And Plasmodium, which can cause malaria, right, by interacting with our red blood cells. Amoebas are other protists, and these, given the fact that their cytoskeletons are different, they're kind of weird weirdly shaped or unicellular bacteria, right? This can also cause very serious illnesses, these amoebas, right? Slime molds are others. They resemble fungi, but again, they're protists. Algae can also be they're photosynthetic, right? So algae are mostly aquatic, unicellular, but also multicellular organisms, right? And they're green, right? What does that mean? Well, that means that they contain the pigment called chlorophyll. If they contain chlorophyll, then that means that they're innately photosynthetic, because they're naturally photosynthetic. So they can create their own food, right? They're autotrophs rather than being heterotrophs, okay? So just a quick little timeline in terms of the evolution of life here on Earth, starting off 4.6 billion years ago, right? I know I'm showing you guys this timeline in millions of years, but 4,600 million years ago means 4.6 billion years ago, right? So that's when Earth originated, right? Then 3,500 or 3.5 billion years ago was the origin of prokaryotes. So it took about a billion years for prokaryotes to be formed. And then from there, 2.1 billion years ago, that was the origin of eukaryotes, right? So it took about what? Another 1.4, give or take, billion years for eukaryotes to be formed, okay? So look at the time frame, right? We're talking about billions of years. This didn't happen from one year to the next, right? It takes, as I already mentioned, billions of years for life to evolve. At least early forms of life. Nowadays, it takes millions rather than billions, right? Or even thousands, as mentioned in the previous lecture there. So the origin of multicellular life occurred about 1.2 billion years ago. And then animals originated about 600 million years ago. Land terrestrial plants occurred about 490 million years ago. And then humans, actually humans as we know them as we are nowadays, appeared about 300,000 years ago. Okay, so the modern human. But the lineage, right, that gave rise to the modern human appeared about 2.5 million years ago. But there were species before, right, about 8-9 million years before the actual human. So the lineage of the human being, of the Homo genus, if you will, is about 7 to 9 million years old, more or less. Again, there's debate on that, but it's in the area of 7 to 9 million years old. Now, let me differentiate between those and the appearance of humans, right? Humans belong to the Homo genus, okay? And our species is sapiens, right? We're Homo sapiens. So that lineage, right, which took a few actually different species, like Homo erectus, Homo habilis, for example, right? And Homo neanderthals, among others, out of which we actually emerged from the Neanderthals, from different populations of the Neanderthal species, right, which was in existence right before us. But it turns out that we actually lived right along them, right? So imagine seeing other species very similar to us, but that are not quite human, but look humanish, if you will, right? Neanderthals, again, they were human-like species, but their brains were smaller. Their heads were a little bit smaller. So the difference between them and us is that our brains have more volume, right? We have a larger skull, therefore a larger brain volume. Therefore, more connections could be made, more brain tissue, more neurons, and so on. Okay, so, but regardless, we have Neanderthal genetics. Okay, so if you do a little bit of research into Neanderthal and human genetics, you'll find that a lot of our immune system actually came from Neanderthals. Okay, so pretty interesting stuff, at least in my view. Okay, so some of the last concepts here in this lecture are viruses. Okay, we've already touched on viruses. Viruses are not alive, they are not cells, but they do contain genetic information encapsulated in a capsid, right, which is made up of capsomeres, right? So these little units here, these are capsomere proteins, which if they're placed in the right shape, in the right way, capsomeres actually make a capsid, right? And the capsid is a capsule which encapsulates, I'm mentioning kind of redundant there, but it protects DNA essentially, right? So nucleic acid is inside, and I'm saying nucleic acid because it can either be DNA or RNA. In the case of HIV, HIV contains RNA. Okay, but keep in mind the other, right? The last part of the virus here is the glycoprotein, otherwise known as the recognition spike, quote unquote, if you will, right? So glycoprotein that allows viruses to attach to their hosts. Okay, so once in hosts, right? Why would they even infect hosts, right? But well, because they need to reproduce, they need to make more of themselves. That's essentially why they have to use other host cells, right? So once they manage to do that, then they're gonna go ahead and go through two life cycles. One's called the lytic cycle, the other one's called the lysogenic cycle, right? The lytic cycle is more of a kind of quick type of reproduction type of thing, if you will, right? Which allows viruses, as soon as they dock onto the host cell, they can go ahead and make a lot of copies of themselves within days, okay, within weeks, if you will. All right. So now lysogenic is more of embedding DNA into the host cell's DNA type of thing, and sometime at a later point in time, the virus can actually become lytic, right, and it can cause disease and make many copies of themselves, right? But after months/years of infection, okay. Like HIV, for example, HIV. Most of the time, I'm not saying every single patient, right? HIV takes years, right? Months to years to incubate, if you will, right? So the time from infection to the time where the disease starts to manifest, right, in symptoms, that's the incubation period right there. AIDS has a very long incubation period, from months to years, right? So a person with an infection will not be able to know that they have DNA until, may not know within years, right, that they have HIV, unless obviously they find out, they suspect that their partner had HIV. So in that case, they go ahead and carry on with an HIV test, and if it comes out positive, then evidently they're infected with HIV, right? HIV stands for human immunodeficiency virus, whereas AIDS, right, is the acquired immunodeficiency syndrome. So HIV is a virus that causes AIDS over time. But again, AIDS is a syndrome, meaning that it's a collection of many different symptoms, right? So, and it's caused by HIV over years of infection, or probably months of infection, right? Depending on the the patient's immune system, right? So HIV is a retrovirus, right? Meaning that it contains RNA naturally. And once inside the host, it must make DNA from RNA by using this enzyme called reverse transcriptase. Okay, so this is the life cycle of HIV. It's more of a lysogenic type of virus, binding, right, to the host cell, evidently after sexual contact. The virus, after docking, right, after docking onto the cell of the host cell, then phage DNA or viral DNA is going to be injected, right? So as that DNA is injected into the whole cell, then or RNA rather, that RNA has to become DNA, right? Again, the action of reverse transcriptase takes care of that. Reverse transcriptase makes RNA into DNA, right? So once DNA is made, then it can become embedded onto the whole cell's DNA, and it can remain there for months, years, right? Just kind of making a lot of copies of that. And then once it becomes, ideal, if you will, for this phage DNA to divide, then it's gonna go ahead and become a lytic, if you will, type of virus at that point, and that can happen because of many reasons, including stressful situations, environmental conditions, and so on, or just naturally, right? So it'll become a lytic virus. It'll start making many copies of itself, and then causing all sorts of symptoms after that. Now, there's differences between different viruses, and in terms of how they make copies, right, of themselves. HIV, it turns out that HIV has to actually attain a piece of the membrane of the whole cell, and in that manner, right, they can actually leave, right? They can be exocytosed from the whole cell, right? So again, the process of creating, if you will, these new baby DNA, baby viruses, if you will. Those are called virions, by the way, right? So that process is called the incubation period. And it might take, for HIV, might take anywhere from months to a few years to achieve. Okay, so mostly lysogenic, but again, ultimately, all viruses need to come back, if you will, to return to a lytic stage in order to make many copies of themselves. All right, so now keep in mind HIV is mostly in the immunocompromising type of virus, meaning that it's going to go ahead and attack T cells from our immune system. So given that fact, there's different types of T cells. One of which is called the CD4 helper T cell. HIV is highly specific for those CD4 helper T cells, and if they become damaged by the HIV virus or destroyed by the HIV virus, then they are rendered useless, right? Making the patient immunocompromised. Therefore, the patient develops into acquired immunodeficiency syndrome, which at which point they can actually suffer from other infections and not being able to fight off other infections, which is for the most part how HIV/AIDS patients end up dying, right, from other infections, if you will. Okay, so now let me touch on other types of pathogens. Those are called prions, right? So prions are actually misshapen infectious proteins, right? So some and even pieces, right, of proteins that were not shaped correctly. So this is the reason why our cells must achieve the correct folding, right, of proteins into all of their structures: primary, secondary, tertiary, and even quaternary, right? If there's issues, then we have physiological mechanisms like proteasomes that allow us to get rid of proteins that are misshapen, right? But then sometimes, and rarely, right, These misshapen proteins are not dealt with, therefore they can cause infections, and those are called prions, by the way. Alright, so that was the last topic of the lecture. If there's any questions, be sure to send them to me through email or request a virtual meeting.